students, welcome to day 15 of PU2 Brahmastra Series, and today is Monday. It's time to study chemistry, right? So today we are going to do one very easy and scoring topic, that is biomolecules. So I'll be discussing all the possible questions from this chapter, and if you do these many questions, then definitely you can answer all the questions being asked from this particular chapter. I can guarantee you that. So students, what you need to do is sit with a notebook, note down the questions, try to answer them. If you are not able to answer, then please see the solutions, write down the answers, and remember these questions, learn these questions. Okay, so let's get started here. And one more request, please join our WhatsApp channel. Link is there in the description section. You will be getting access to all these notes and everything in our WhatsApp channel. Okay, so please join the WhatsApp channel if you want the PDF notes of these classes. Okay, so now coming to the weightage of this chapter, it is having more weightage, that is 9 marks will be asked from this particular chapter. There will be 2 very short answer questions, 1 2-marker questions and 1 5-marker question. Okay, so 5, 6, 7, 8 and 9 marks you will get. Now coming to, in this particular lecture we will be discussing 10 VSA. Okay, all these questions are of HOTS level. Okay, then 2-marker questions I have taken 34 questions, and 5-marker questions I have taken 2. Okay, now a question may arise like, why only 2 5-marker questions? Now see, whatever I have taken in 2 markers, right? So if these 2 questions are mixed, like suppose some questions like these different questions are mixed from one point and asked in the 5-marker question. So if you do these 34 2-marker questions, you can easily answer the 5-marker questions. The combination of 2 markers questions will give you the 5-marker question. Okay, so need not worry about that at all. And now let's get started, my dear students. Okay, so coming to the first question here. Okay, select the correct statement regarding amylopectin. So what is amylopectin? Let us discuss small thing. So see, first is carbohydrates. Okay, under carbohydrates we have monosaccharides, disaccharides, right? How many monomer units are there? So if more number of monomer units are there, we call it polysaccharides. So under carbohydrates we have polysaccharides. Okay, under polysaccharides we have one example is starch, and starch is made up of 2 things, that is amylose and amylopectin. Okay, so amylopectin is a branched polymer of alpha D glucose. Okay, it has C1C4 linkage and C1C6 linkage. Okay, means carbon glucose units are there, C1C4 linkages are there, C1C6 linkage is there, right? So now I think you can answer this question. Amylopectin is a polymer of alpha D glucose units, right? I told you it is a polymer of alpha D glucose units, so this statement is correct. It constitutes about 15 to 20% of the starch. Now here see, amylose consists of 15 to 20% and rest, that is 80 to 75%, sorry, 80 to 85% are from amylopectin. So this statement is wrong. It is water insoluble component of the starch. Yes, this is also correct. Here I'll tell you, amylose is water soluble. Okay, water soluble, whereas amylopectin is water insoluble. Clear? So I can say statement 1 and 3 are correct. So option number A is the correct answer here. Clear? Next question. The number of amino acids in tetrapeptide. Now see, what is peptide? If you have, so proteins. Okay, next we have biomolecule is protein. Protein is made up of amino acids. Okay, proteins are made up of amino acids. Amino acids are linked by, so we have amino acids, these are all amino acids, they are linked by peptide bonds. Okay, so in a tetrapeptide we have 4 amino acids. Okay, so that is why option number C is the correct answer here. Okay, next question number 3, which one of the following is true regarding lactose? Okay, so what is lactose? Again we will see, we have carbohydrates, right? We have carbohydrates, under that we have disaccharides. Okay, monosaccharides, disaccharides. Under disaccharides, one of the example is lactose. Its monomer units are glucose, okay, and galactose. Okay, Now let us see the question here. You have to find the not correct statement. Okay, be very careful. Not correct statement. Okay, on hydrolysis, lactose gives equal amount of glucose and galactose. Yes, this is correct because it is made up of glucose and galactose. So if you break lactose, you will get glucose and galactose. This statement is correct. Now lactose is a beta glucoside formed by the union of a molecule of D-glucose and a molecule of D-galactose. Yes, this statement is also correct, right? Now lactose is a reducing sugar and does not exhibit mutarotation. This statement is incorrect. Now see, reducing sugar means what? Any sugar which is having free aldehyde group. Okay. Free aldehyde group. Okay. So lactose is a reducing sugar and it has a free aldehyde group, and anything which has a free aldehyde group will exhibit mutarotation. Mutarotation means they can change their optical activity. Okay, their specific rotation can change. Okay, so that is why this is the incorrect statement. Option number C is the correct answer. Next, question number 4, which one of the following statements is not correct regarding the similarity between glucose and fructose? So again, if I see carbohydrates, okay, we'll go back, carbohydrates, okay, under carbohydrates we have monosaccharides, okay, monosaccharides, and under monosaccharides we have two examples. One is glucose. There are many more, but these are the things which we studied in detail. Glucose and fructose. Glucose has a functional group of aldehyde, fructose has a functional group of ketone. Okay, that is the difference between them. Okay, which of the following statements is not correct regarding the similarity between glucose and fructose? Okay, fine. Both give pentaacetyl derivatives with acetic anhydride. That is correct because both of them have 5 OH groups. Okay. Next, with HCN both give cyanohydrin. That is also correct. You know aldehydes and ketones both will react with HCN to give you cyanohydrin. So that is also correct. Now with phenylhydrazine both give similar ozazone. That is also correct. Now both contain aldehyde functional group. No, you know fructose contain ketone functional group. So that is why this statement is incorrect. Option number D is the correct answer here. Next, question number 5. In both DNA and RNA. So now coming to another biomolecules, that is nucleic acid, right? So under nucleic acid we have two types. One is your RNA and another is your DNA. Okay, the question is about that. So the heterocyclic base and phosphate ester linkages are at. Okay, now see, as I told you, carbohydrates are the polymers of monosaccharides, okay, proteins are the polymers of amino acids. Similarly, nucleic acids are the polymers of nucleotides, okay. Nucleotides comprises of 3 things: sugar, nitrogenous base and phosphate group, okay. So sugar is a 5-membered ring. On the first carbon, okay, we have a nitrogenous base here. One second. We have nitrogenous base here, okay, on the first carbon, and 1, 2, 3, 4 and 5. So now on this particular 5th carbon, okay, and here we have oxygen. So 1, 2, 3, 4 and 5, we have a phosphate group, okay. So carbon 1 has base and carbon 5 has the phosphate group. So you can say heterocyclic base is present on C1 and the phosphate group is present on C5. So that is option number C here of the sugar, and this whole group is called as your nucleotide. Clear? And these many nucleotides together will comprise one nucleic acids. Okay. Fine. Next, question number 6. Select the incorrect statement from the following. Okay. Glucose exhibits in 2 different crystalline forms, alpha glucose and beta glucose. Yes, that is correct. Okay. Now, alpha D glucose and beta D glucose are enantiomers. No. Enantiomers are different thing. Alpha D glucose and beta D glucose are anomers. Okay. Now, what is anomers? We will be solving questions based on that on the 2-marker question. But I will say what is anomers means the entire configuration will be same, but only at C1, only at 1 carbon the configuration will be opposite. Okay, only the configuration will be opposite at the first carbon, and that is called as anomers. Clear? Next, Okay, cellulose is a straight chain polysaccharide made up of only glucose units, that is also correct. Starch is a mixture of amylose and amylopectin, both contain unbranched chain of alpha D glucose units. This is also wrong. Why? Because amylopectin contains unbranched chains also, right? In amylopectin you have C1 C4 linkage and C1 C6 linkage, right? This is unbranched, this is unbranched, whereas C1 C6 is branched lane. Clear? So what you need to tell? Okay, select the incorrect statements from the following. So which two are incorrect? 2 and 4. Okay, so we can say 2 and 4 are incorrect but not 1 and 3. So option number C is the correct answer here. Okay, option number C. Okay, next question number 7. Which of the following statements is incorrect? So it is about all in total. So proteins have a polyamide chain while nucleic acids contain a polynucleotide chain. Yes, this is correct. Nucleic acids are long chain polymers of nucleotide, that is also correct. RNA is a major source of genetic information which is copied into DNA molecule. No, DNA is the major source of genetic information. Okay, the incorrect statement is option number C here. Next question number 8. Which of the following groups include all essential amino acids for, okay. Now here I will be telling you what I can say, a mnemonic to remember the essential amino acids. So first of all you should know that there are 9 essential amino acids and the mnemonic that follows here is PVT. Okay, then we have T, one second, okay, HILL TIM. Okay, so I think you can say like private team hill. Okay, something like that, private team hill. L I'll put as capital. Okay, now P is your phenylaniline. Okay, V is valine. Okay, T is threonine. This T is tryptophan. Then you have isoleucine. Okay, then you have methionine here, then H stands for histidine. Okay, L both are for lysine and leucine. Okay, so I can show you the list of these amino acids here. So you have valine, leucine, isoleucine. Sorry, here are 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah, this arginine is not there. Okay, then we have lysine, threonine, methionine, phenylalanine, tryptophan and histidine. Okay, so you have to select all the essential amino acids from here. So if you see, we have tryptophan, phenylalanine, glycine here is not there, we can eliminate this option. Leucine, methionine, lysine and valine, yes, all are there, option number A. Here glutamine is not there, eliminate that. Phenylalanine, valine, lysine and tyrosine is not there, so we can eliminate that. Okay. Next, question number 9. The alpha and beta forms of glucose are isomers of D-glucose and L-glucose respectively. No, they are the isomers of D-glucose only. Diastereomers of glucose? No, they are anomers of glucose. Yes, option number C is the correct answer here. Next question number here, thymine and pyridoxine are also respectively known as. So these are the chemical names of vitamins. So thymine is your vitamin B, okay, sorry, vitamin B1 and pyridoxine is your vitamin B6. Option number A is the correct answer here. Okay, so now we are done with the BSA or you can say MCQs. Now we will move towards 2 marker questions. So first we will start with the questions of carbohydrates. So first is, what are monosaccharides? Give examples. So as I told you, carbohydrates are made up of small units. Okay, the monomers of carbohydrates are monosaccharides. So when you break a carbohydrate, if you are getting 2 monomer units in that, then it is a disaccharide. If you get 3, then you get trisaccharide. But carbohydrates which cannot be further hydrolyzed, the simplest unit of carbohydrates are called as monosaccharides. Okay, so example you can see, like answer, they are the simplest carbohydrates and cannot be further hydrolyzed. You cannot break a monosaccharide, right? So that is what is called as monosaccharide. Examples: glucose, fructose, galactose, ribose, deoxyribose. You can just remember glucose and fructose. Clear? What is monosaccharide? Monosaccharides are the units of carbohydrates which cannot be further hydrolyzed. Okay. Next question is, what are disaccharides? Give an example. So disaccharides, from the name you can see di is there. That means when you break that or when you hydrolyze the disaccharides, you get 2 monosaccharide units. Okay. So carbohydrates on hydrolysis gives 2 to 10 monosaccharides. These are called as, okay, one second. Yeah. Now so you need not write these many things answered here. Okay. For example, you can write down. Okay, let me give the answer for this one here. If hydrolysis, okay, of a carbohydrate, if a hydrolysis of a carbohydrate gives 2 monosaccharide units, it is called as a disaccharide. It is a disaccharide. Okay. Example you can give here. Okay. For example, this is sucrose, right? When sucrose is undergoing hydrolysis, you are getting 2 units. One is glucose and another is fructose, right? Similarly, if you have maltose, okay, when maltose undergoes hydrolysis, you get 2 units of glucose. Okay. Next, lactose. So if lactose undergoes hydrolysis, you will get 1 glucose and 1 galactose. Okay. So when you break this, you are getting 2 monosaccharide units, that is called as disaccharides. Okay. Now what is oligosaccharide here? That also you should know. So oligosaccharide is a broader category. If you hydrolyze any carbohydrate and you are getting 2 monosaccharides minimum or at least 10 maximum monosaccharide units, these all carbohydrates comes under the category of oligosaccharides. If you are getting more than 10 monomer units, then it comes under the category of polysaccharides. Okay. Monosaccharides you cannot further hydrolyze. Oligosaccharides, on hydrolysis you are getting 2 to 10 monosaccharides, and if you are getting 2, it is a disaccharide. If you are getting 3, it is a trisaccharide. Clear? Next, question number 3. Give an example of homodisaccharide and heterodisaccharide. Homodisaccharide means what? When you break, okay, you get 2 same monomer units, and disaccharide means when you break, you get 2 different monosaccharide units. Okay, this is very simple. So maltose is your monosaccharide. Okay, sorry, maltose is homodisaccharide. Okay. Now the reason for that is when you break maltose, you get glucose and glucose. But there is also an example of hetero, right? So for hetero, you can take sucrose. Okay, sucrose plus water will give you glucose and fructose. Their formula is same, but the 2 units are completely different. Okay, this is heterodisaccharide. Question number 4 here. What are reducing sugars? Give example. This is very, very important. Okay. So reducing sugars are those sugars which can react with Tollens reagent or Fellings reagent. Now how to know whether they will react or not? If they have a free aldehyde group. Okay. So now you can see the carbohydrates which reduces the Fellings solution and Tollens reagent are called as reducing sugars. Example: glucose, galactose, fructose, maltose, lactose. Okay. Now why they can reduce Fellings solutions and Tollens solution? Because they have a free aldehyde group or a keto group adjacent to the CHOH group. Okay. So if they have a free aldehyde group, then they can reduce the Tollens reagent and Fellings reagent, and then we can call them as reducing sugars. Okay. Next, question number 5. What is non-reducing sugar? Give an example, very opposite to that. So here the carbohydrates which does not reduce the Fellings solution and Tollens reagent due to the absence of aldehyde group and keto group are called as non-reducing sugar. Example is sucrose. So you should be very, very particular about the examples here. Clear? Next, question number 6. Which of the following is called as grape sugar? So grape sugar is your glucose, Okay, fruit sugar is fructose because it is found in fruits. Cane sugar is sucrose because it is found in sugar cane. Malt sugar is maltose. Okay, milk sugar is lactose because it is found in milk. Invert sugar is again sucrose. Okay, clear? Next, give the chemical names of the following sugar. Okay, so glucose is called as dextrose because it is dextrarotatory. Okay, fructose is called as levulose. Levulose, one second. Levulose. Levulose. I am little confused with the spelling. You can check the spelling, students. Okay, levulose. And sucrose is called as saccharide. Okay, these are the chemical name for these particular sugars. Next, give two methods of preparation of glucose. So, glucose can be prepared, see, glucose is the monomer unit of sucrose. So, if you break sucrose, you can get glucose, right? That is one of the method, and let's see the other method here. So, first method is from sucrose, okay? That is your cane sugar. Sucrose is boiled with dilute HCl or H2SO4 in alcoholic medium to give you glucose and fructose in equal amount, okay? So, this is sucrose. It is boiled in acidic medium and we get glucose and fructose in equal amount. Clear? Next, from starch, okay, so we can also prepare it using starch molecules. So, glucose is obtained from commercially by boiling starch with dilute H2SO4 at 393 Kelvin under pressure. Okay, so this is the formula from where we can get from starch. Okay, so I'll clear this so that it is not looking messy. Okay, okay. Let us move to the next question here. How does glucose react with hydrogen iodide, or how do you show that glucose contains 6 carbon atoms in a straight chain? Okay, so now you see here, glucose, to understand the structure of glucose, there are 6 reactions. That 6 reactions are very, very important. We will do those 6 reactions in the form of questions. Okay, so the first thing here is, first conclusion that glucose has 6 carbon atoms and all the 6 carbon atoms are present in one chain. Okay, that conclusion was given by the reaction with HI. Okay, so the question can be asked in either way. How does glucose react with hydrogen iodide, or how do you show that glucose contains 6 carbon atoms in a straight chain? So, if this is asked, how do you show that glucose contains 6 carbon atoms in a straight chain? The answer is by reacting it with HI. So, what is the reaction here? You can see glucose on heating with HI and red phosphorus gives N-hexane. This shows that glucose contains 6 carbon atoms in a straight chain, and this is the reaction. Clear? The first reaction of glucose was with HI and it will give you N-hexane as the product. The conclusion that we get here is that glucose has 6 carbon atoms in one chain. Okay. Now, let us go to the second question here. How does glucose react with hydroxylamine NH2OH, or how do you show that glucose contains carbonyl group? Okay, so this is the second reaction of glucose to show the presence of carbonyl group. It reacts with hydroxylamine. Okay, so you can see that glucose reacts with hydroxylamine to give glucose oxime. This shows the presence of a carbonyl group. Okay, so reaction you can see here, you have CHOH whole 4 glucose with NH2OH will give you this one. Okay, so this is your glucose oxime. This reaction is very much similar to you in the aldehydes, ketones and carboxylic acid chapter. Here H2 and O will be removed, CH double bond NOH will get. This is your oxime group. Okay, so I told you there are 6 reactions of glucose that you should know. The first one is reaction with HI, and what was the product there? N-hexane. What conclusion we got was about the straight, or the 6 carbons in the glucose are in a straight chain, right? Second thing was about, the second reaction was about with NH2OH. Okay, the product here was glucose oxime, and the conclusion that we got here was that glucose has a carbonyl group. It can be either aldehyde or ketone, but we know that it is having one carbonyl group. Okay. Next, third reaction, how does glucose react with acidic anhydride, or how do you show that glucose contains 5-OH group? Okay, this is the second reaction here. So you can see glucose reacts with acetic anhydride, gives glucose pentaacetate, which confirms the presence of 5-OH group. Okay, so glucose, if you react with acetic anhydride, we get a pentaacetate. Okay, so when means alcohols on reacting with anhydride giving you esters. So if you are getting 5 pentaacetate group or 5 ester group, that automatically means that there will be 5-OH group here. Okay, next thing, how does glucose react with bromine water, or how do you show that glucose contains an aldehyde group? Okay, so here is this important. See, first reaction showed that glucose has 6 carbon, one second, yeah, here the first reaction shows that glucose has 6 carbon in a straight chain. Second reaction of glucose shows that it has a carbonyl group. Okay, the third reaction of glucose is actually this one. The third reaction of glucose with Br2 shows that it has aldehyde group. Okay, fourth reaction of glucose is with this pentaacetate, okay, which shows that it has 5-OH groups. Okay, and then the fifth reaction of glucose is with, we'll talk about that in the next question. Okay, for now I have only told you this many. Now how does it reacts with bromine water? So now see, bromine water is a mild oxidizing agent. Okay, it is a mild oxidizing agent. So what it will do, if it is an, if we don't know whether it is an aldehyde group or a ketone group, so we are thinking, okay, let us react it with bromine water. Okay, so if bromine water, if it is an aldehyde, then it will get converted to acid, because aldehydes can be oxidized to acids, but ketones will not be further oxidized, right? So based on that, we are just guessing whether it is aldehyde or ketone, and we reacted it with bromine water, and we found that one acid group was formed, and that confirms the presence of an aldehyde group. Okay, so that is here. You can see here this CHO group got converted to COOH group. Okay, now why these all things do not get converted? Because these are all your, these will be get converted on a strong oxidizing agent, right? These are all mild oxidizing agent which can only convert aldehyde to acids. If this would have been ketone, then we would not have gotten acid here, right? So glucose on mild oxidation with bromine water gives gluconic acid. Okay, this is your gluconic acid. This indicates the carbonyl group present in glucose is an aldehyde group. Okay, so again let us revise. First reaction is with HI, which tells you that there are 6 carbons in one chain. Second reaction is with NH2OH, which tells you the presence of a carbonyl group. The third reaction is with bromine water, which tells you the presence of aldehyde group. The fourth is the reaction with an anhydride, which tells you the presence of 5-OH groups. Okay, now we'll see the next one here. How do you show the presence of alcoholic group in the glucose? Okay, so okay, now you see here, if you react this one, okay, so this is actually how do you show the presence of alcoholic group OH in the glucose. Now see, on oxidation with nitric acid, okay, glucose as well as the gluconic acid yields a dicarboxylic acid or saccharic acid. This indicates the presence of a primary alcoholic group in the glucose. Okay, so we said that there are 5 OH groups, but now how can we show that whether these OH groups are primary or secondary? So what we did is we treated the glucose, okay, with a strong oxidizing agent, that is concentrated HNO3. Now when you treat it with a concentrated HNO3, the primary group will get oxidized to acid, right? So we can find here that glucose, this OH group got oxidized to COOH. Okay, and this, if you take gluconic acid where already we had one COH group, now again if you oxidize that also we got this one. Okay, but these OH were not affected. So that is why we can conclude that out of that 5 OH group there is one secondary, sorry, out of 5 OH group there is 4 secondary and 1 primary OH group. Okay, next question. So what is the open chain structure of glucose? So you have to draw the open chain structure of glucose. So we have 6 carbons, sorry, we have 6, one second, we have 6 carbons here. So first one is CHO group, and here we have CH2OH group. Okay, now you have 1, 2, 3 and 4 carbons. Okay, here this side we have OH, OH, OH and OH. Okay, this is what? This is H, this is H, this is H, this is H. Okay, this is the structure or open chain structure of glucose. Now see, we have something to learn here, that if this OH group, okay, if this OH group is towards the right hand side, okay, then we call it D-glucose. Okay, if this OH group is on the left hand side, then we will call it L-glucose. Okay, capital D, capital L glucose are different than small d, small l. Small d and small l tells about dextrorotatory and levorotatory, whether they will rotate the plane polarized light clockwise or anti-clockwise. But this capital D and capital L tells you about the configuration of this OH, whether it is towards the right side or towards the left hand side. Okay, clear? Now we will go to question number 12. What are the limitations of open chain structure of glucose? Okay, so now glucose can exist in two forms, cyclic form and open chain structure. So the discovery of the cyclic structure took place due to some of the limitations that were found in the open chain structure. So 3 marker question, 2 marker question, you just need to know 3 limitations are there. So the first one you should know is glucose although it contains an aldehyde group, but it does not react with Skiff test and do not react with NaHSO3. Okay, so first point, does not react with Skiff's reagent or NaHSO3. Okay, second point, pentaacetyl glucose, okay, does not react with hydroxylamine, indicating the absence of a free aldehyde group. Pentaacetyl glucose does not react with hydroxylamine. Okay, next, number 3, very important, D-glucose is found to exist in 2 different crystalline forms, that is alpha and beta. Okay, it has 2 different forms, that is alpha and beta. What are the limitations? First one, it does not react with Skiff reagent or NaHSO3. The second one, the pentaacetyl form did not react with you can say hydroxylamine or NH2OH. Third, it existed in 2 different forms, that is alpha and beta. Okay, so any 2 limitations you should remember, it can be asked. Next, question number 3, what are anomers and what are epimers? Okay, now see, anomers means sugars which differ in their configuration at any one carbon is called as epimers. Okay, here a small mistake. Okay, suppose there are 6 carbons in a glucose molecule, they are differing in only 1 carbon. Okay, that is called as epimers. Next, the sugars which different in their configuration at C1 or the first carbons are called as anomers. Okay, if that 1 carbon which is different in two isomers, okay, if 2 isomers A and B are there, their configuration, suppose, now let us take an example here, okay, A and B. Okay, so we have one structure like this A and another structure like B. Okay, this is carbon 1, carbon 2, 3, 4, 5 and 6, 1, 2, 3, 4, 5 and 6. Okay, now A and B will be called as epimers, okay, if they have difference in any 1 carbon, configuration will be different in any 1 carbon. Now the epimers can be also called as anomers, okay, if that 1 carbon where the configuration is different, that is your C1, that is carbon number 1. So if here the configuration is different, okay, then it will be an epimer. Okay, but if the configuration is different on the first carbon, okay, then it is an anomer. Okay, or you can say anomer is a subset of epimers. Okay, and all anomers will definitely be epimers, but all epimers cannot be anomers, because it can be at any, if configuration 5 is different, then also they will be epimers, but they won't be anomers. Okay, I hope this is clear. Next question. Okay, write the Haworth structure of beta D glucose and alpha D glucose. Okay, so we have seen that there were some limitations of the open chain structure. So due to that, we also have the cyclic structure of glucose, and we have 2 projections of cyclic structure. One is alpha glucose, one is beta glucose, and these are called as anomers. Now you see, this carbon, okay, carbon number 2 configuration is same, carbon number 3 configuration is same, 4th configuration is same, 5th also configuration is same. In the first carbon, here OH is below, and in the first carbon OH is up, right? So the one where OH is below is called as alpha carbon, and the one in which OH is above is called as beta glucose. Okay, so now in only one position the configurations are different, so that is why we can call that anomers. They are anomers. Okay, clear? These are called as anomers. Okay. Next, so now why this D is written, all of you understood? D is because that OH was in the open chain structure, OH is towards the right hand side. This plus shows that it is dextro-rotated, it means they will rotate the plane polarized light in clockwise direction, okay, and alpha tells you about this configuration, okay, fine. So now let us go to the next question, number 15. Write the Haworth structure of alpha D maltose. Okay, so now we are done with the monosaccharides and we will go towards maltose, okay, sorry, disaccharides. Under disaccharides we have 3 disaccharides, that is maltose, sucrose and lactose, okay. For all these 3, maltose, lactose, sucrose, you should know their monomer units and the linkage between them, how the monomer units are linked between them, okay, that is very, very important, and whether it is a reducing sugar or a non-reducing sugar, okay, that is again important. So what is the Haworth structure of maltose? You should know this drawing, it is a little complicated, but yes, you have to practice this one. So in maltose what happens? We have alpha D glucose, the monomer units are alpha D glucose. So you can see this was one, if you see the structure of alpha D glucose, now let me draw and show you. Suppose we have alpha D glucose structure is like this, alpha, right, OH will be at bottom, here OH is at bottom, here it is H, here it is OH, here it is H, here it is OH, here it is H, and here it is H, CH2OH, okay, and we have another structure like this, okay, and this carbon, if you see here also OH is there. So now this is C1 and this is C4, okay, one C1 and one C4, alpha D glucose units, there is what you can say, this bond is glycosidic bond and there is a loss of water molecule, and that is why it is visible like this, okay. So we have the monomer units of alpha D glucose, here carbon 1, carbon 4, glycosidic linkage is there. So when 2 monosaccharides are joined, that bond is called as glycosidic linkage, okay. So now question, mention the type of glycosidic linkage. So that is C1 C4 glycosidic linkage, okay. What is the type? C1 of one glucose is linked with the C4 of another, okay. Next, give the reason maltose is a reducing sugar. Now see, so the answer here will be that maltose has a free aldehyde group, that is why it is a reducing sugar, okay. So how does that free aldehyde group comes? Here we can do tautomer, like here the rearrangement can change like this, okay. This one can come here and this bond can break and we will get a free aldehyde group here, okay, that is possible. But how to know, what is the trick to find out that it is a reducing sugar or not? First let us talk about the answer. So the free aldehyde group can be produced at C1 of one of the glucose in the solution, so it is a reducing sugar. But how to find out, what is the trick? So the trick here is you need to search for hemiacetal group. You need to search for hemiacetal group at C1. Okay, hemiacetal group, if you remember, COH and OR group if it is present. If we have hemiacetal group, one OH and one OR attached to one carbon, then we can call it as a reducing sugar, okay. But you do not have to write that in the answer. Answer is it can produce a free aldehyde group. Now if you see hemiacetal group at C1, this is C1, right? Here you see this is OR and this side also it is OR, there is no OH group here. So this carbon is not there. But if you see this carbon, right, it has a OH group and this side it has a OR group, right? Because it is having an hemiacetal group, it will be behaving as a reducing sugar. Clear? So maltose is done. Next we'll go to lactose. Okay, so lactose structures you can see here, it is beta D galactose and beta D glucose. Again C1 C4 linkage is there. Okay, mention the type of glycosidic bond. It is between C1 of beta D glucose and C4 of beta D glucose. C1 of galactose and C4 of beta D glucose. Next, why it is a reducing sugar? Because it has a free aldehyde group. Again you can see OH and OR group is present here. Okay. Next question number next question number 17, name the storage polysaccharide present in animals. So now we are moving towards polysaccharides. Okay, so disaccharides, we have three types of disaccharides, that is sucrose, lactose and maltose. Sucrose I have not taken any question, I will be taking it in the 5 marker questions. Okay, and polysaccharides also we have three types of polysaccharides, that is your starch, cellulose and glycogen. Okay, starch, cellulose and glycogen. So first it is question is asked, name the storage polysaccharide present in animals. Where is it found? So what I am saying, let me first write down here polysaccharides. We will just have a quick revision here. Polysaccharides we have three types. Okay, one is starch, then we have glycogen, okay, and another is cellulose. Okay, glycogen is found in animals. Okay, cellulose is found in plants. Okay, starch comprises of two things that we have already discussed. One is amylose and amylopectin. Okay, next glycogen structure is similar to amylopectin. Okay, in cellulose the monosaccharide units is beta D glucose units. Okay, and in starch and this one it is alpha D glucose units. Okay, here also it is alpha D glucose units. Okay, this is all about polysaccharides. Now the storage polysaccharide present in animals is your glycogen, and it is found in liver and muscles and brain. Okay. Next question, give any two difference between starch and cellulose. Okay, so you can see starch is made up of alpha D glucose and cellulose is made up of beta D glucose. Okay, now starch has what type of linkage? It has alpha glycosidic linkage, it has beta glycosidic linkage. In starch there is both C1 C4 and C1 C6 linkage. In cellulose it is only a linear polymer, you have C1 C4 linkage. Okay, next you have linear and branched polymer, it is only a linear polymer. Okay, so here I can write that what I mentioned, that cellulose is linear. Okay, starch has both linear plus branch structure. Okay, so difference here I think it is clear. Starch is made up of alpha D glucose and cellulose is made up of beta D glucose. So here it is alpha glycosidic linkage, here it is beta glycosidic linkage. Starch has both linear and branched polymers. Linear is having C1 C4, branched is having C1 C6. Here it is only linear with C1 C4 glycosidic linkage. Okay. Next question number 19, we will now go towards the proteins. Okay, then second biomolecule, that is proteins. Now what are alpha amino acid? Write the general formula of alpha amino acid. So amino acids are groups where you have one NH2 group and one COOH group. Okay, so you can see here alpha amino acids are compounds with amino group and carboxylic acid group. Okay, there is one amino group and there is one carboxylic acid group. So what is the structure of this one? You have to draw the structure also. So general formula, you have one COOH group, one NH2 group, one H group and one R group. Okay, so based on these R group we have total 20 amino acids. Okay, clear? Next question, what are essential amino acids? Give one example. This is very important question. Now see, amino acids which cannot be produced in the body. Okay, there are certain amino acids which cannot be produced in the body, and because that is required for our health, we have to consume it from the diet. Okay, those type of amino acids are called as essential amino acids. And to remember that, I have already told you a mnemonic PVT, right? That is phenylalanine, valine and threonine. Then a TMI, right? Sorry, TIM. So again that is tryptophan, then you have isoleucine and methionine, okay? HILL, HI for histidine, L for leucine and L for lysine, okay? So, and what are non-essential amino acids? Just the opposite of that, which can be produced in the body and need not be taken from outside. So, apart from these 9 essential amino acids, all other amino acids are non-essential amino acid, okay? Example you can see, cannot be synthesized in the body, have to be consumed from diet. This is your essential amino acids, examples are there. Non-essential will be opposite to that, okay? Next question number, okay, can be synthesized in the human body, example are glycine, alanine, serine, okay. Next question number 21, what are acidic amino acids, give an example, or basic amino acids, give example, neutral amino acids, give example, explain the optical activity of amino acids and name the optically inactive amino acid, okay. These all questions can be asked, so I have taken them together. Now see, I told you that in an amino acid there is one NH2 group, there is one COOH group and there is one R group, okay. So NH2 is a basic thing and COOH is an acidic thing, right. So if there are more than one COOH group in that R part, if we have some COOH group, then it will be an acidic amino acid. If in that R part we have NH2 group, then we will be calling it as a basic amino acid. If there is equal number of NH2 and COOH group, then it is a neutral amino acid, okay. So you can see here more number of COOH group than NH2 group, example aspartic acid and glutamic acid. Next, for basic amino acid, more number of NH2 group than COOH group, example lysine, arginine, histidine. Next, equal number of COOH and NH2 group, glycine, alanine and valine, okay. These are the examples you should remember. Next is talk about the optical activity of amino acid. Now you see amino acid, carbon is there, one NH2 group, one COOH group, one R group and one H group. All the 4 groups are different, so that is why they are optically active, right. So amino acids are optically active due to the presence of chiral carbon. But glycine structure, if you see, okay, glycine is an amino acid whose structure is like this, NH2 COOH, we have H here and H here, right. Now 2 H are present on the same carbon, right. So there are not 4 different groups, so that is why this is an optically inactive amino acid, okay. Next question, what how is peptide bond or peptide linkage, how is it formed? So when 2 amino acids are there, okay, they are linked by like 2 monosaccharide units are linked by which units? You can say 2 monosaccharide units are linked by a glycosidic bond. Similarly, 2 amino acids will be linked by a peptide bond, okay. So that is what the CONH bond between 2 amino acids is called as peptide linkage, formed with the loss of water molecule, okay. Now if you want to show, you can draw it and show the structure also. So we have like this NH2, okay, CRH and we have COOH, okay. This is first amino acid and here we have NH2, C, R2, H and COOH, okay. So now what happens here, there is a loss of water from this one, okay, and we get something like this NH2 CRH C double bond O, okay, and then we have NH CR2 H and COOH, okay. Now this bond, this is called as your peptide bond or amide linkage, okay. Next question number 23, write the formula to calculate the number of peptide bonds, okay. What are polypeptides? Now number of peptide bonds formula is n minus 1, okay. The number of peptide bonds is n minus 1, where n is the number of amino acids. So for example, if I have a tripeptide, okay, tripeptide, that means I have 3 amino acids, okay. But how many bonds I need to connect them? I need only 2 bonds to connect with them, right. So if number of amino acid is 3, number of peptide bonds will be 3 minus 1, that is 2. Next, what are polypeptides? So if more number of amino acids have combined, okay, they are called as polypeptide. So polypeptide in a molecule contains more than 10 amino acids joined together by peptide bonds. Okay, example is insulin. When you have more than 10 amino acids connected to each other by peptide bond, that is called as polypeptide. Like we had polysaccharides, it is polypeptides. Okay. Next, question number 24, what are Zwitter or dipolar ions? This is also very important question. Okay. Now, if you see Zwitter ion in a aqueous solution, okay, the COOH group donates a proton. Why? Because it is acidic in nature, and the amino group accepts the proton, giving rise to a dipolar ion known as Zwitter ion. Let us see the structure here. If you have seen, we have like this COOH, okay. So let me write like this. Yeah. So we have here COOH and we have NH2. We have RH and we have R. Right. Now this is in equilibrium. This one will give you H plus and this one will accept H plus. Okay. So what I'll get here R, C, COO minus, H and NH2, so NH3 plus. Okay, so there is a minus and there is a plus here. So this minus and plus, because of this one, is called as a dipolar ion, or we call it as a Zwitter ion. Okay, clear? Done. Next, question number 25. How are proteins classified on the structure or molecular shape? So based on the shape, there are two types of proteins, that is fibrous proteins and globular proteins. Okay, so we have two types of protein, that is fibrous protein and globular protein. So let us see about their differences. So fibrous protein have fiber like structure, okay, and they are insoluble in water. What about globular protein? They have spherical shape and they are soluble in water. Okay, this question can be asked in the point of difference between also. Okay, so you should know how to write. First point, fibrous structure, spherical structure, soluble, insoluble. So fibrous is insoluble, globular is soluble. Next, in these proteins, the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds. Okay, so the polypeptide chains, so the polypeptide chains run parallel to each other. Okay, and they are held together by which type of bonds? Hydrogen bonds and disulfide bonds. But in globular chain, what happens? The polypeptide chains coil around, okay, coil around each other and are held together by the spherical shape is due to the coiling of the polypeptide chain. So they coil around, okay, and they are also held by hydrogen bonds. Now example of fibrous protein are keratin, which is found in your hair, myosin found in the fibers, collagen is present in your cartilage. Okay. And next, if you see examples of globular protein, you have hemoglobin, albumin, insulin, fibrinogen. Okay, fibrinogen very important, present and is required for the clotting of blood. Okay. Next, we have question number 26. What is secondary structure of protein? Okay, this is also very important. Now see, one second. Yeah, what is the secondary structure of protein? Now what happens? When you have a polypeptide chain, okay, the polypeptide chain coil around to give you secondary structure. They coil around further to give you tertiary structure. Okay, so there is a level of the polypeptide chains, level of proteins, primary, secondary, tertiary and quaternary. So primary is the initial basic structure of a polypeptide chain where different amino acids are linked by the peptide bond. Okay, now what is your secondary structure? So now you see the coiling or folding of a polypeptide chain in a protein as a result of hydrogen bonding. Okay, as a result of hydrogen bonding is called as the secondary structure. Okay, there are two types of secondary structure. One is alpha helix, one is beta pleated. So if your polypeptide chain is undergoing coiling, okay, or they form helical structures, okay, that is called as alpha helix. Example is keratin. And beta pleated structures, if the polypeptide chains are held together side by side and are linked together, you can see in beta pleated structure the polypeptide chains are stretched out and maximum extent and are led side by side, which are held together by intermolecular hydrogen bonds. Okay, So in secondary structure, the polypeptide chain can either coil or can undergo helical structure, that is called alpha helix, or they can be side by side and can undergo bonding between them, and that is called as beta pleated sheets. Okay, so this is about secondary structure. Now coming to what is denaturation of protein and which level of structure remain intact during the denaturation of protein. Okay, so now you see, proteins can function very well when you have certain standard temperature, standard pH values. Okay, now if you change the temperature, if you change the pH value, the bonds that is present between different polypeptide chains get disrupted. Okay, as a result of which the protein loses its activity. Okay, it loses its biological function, and that is what is called as denaturation of protein. Now how to write the answer? This is also very important question. You can see how many times it is repeated. So the loss of biological activity of the protein by heating, if you change the temperature or you change the pH, okay, or you in the presence of any salt, is called as denaturation. Now during denaturation of protein, the primary structure is remaining intact. Okay, so secondary structure will be destroyed, tertiary structure will be destroyed, quaternary structure will also be destroyed, but the primary structure will remain as it is. Okay, next question number 28, what is tertiary and quaternary structure? So if you see, primary structures when it folds it give you tertiary, secondary structure unfolding will give you tertiary structure will give you quaternary. So that is what tertiary structure represents the overall folding and the superimposition of the polypeptide chains. Tertiary structure is stabilized by hydrogen bonds, disulfide linkages, van der Waals forces and electrostatic forces of attraction. Now what is quaternary structure? Now some of the proteins are composed of 2 or more polypeptide chains referred to as subunits. Okay, in quaternary structure, see, you have one polypeptide chain, that is your primary structure. The same polypeptide chain, okay, if it is undergoing coily, okay, secondary structure has 2 things. One is alpha helix, and if remains like this, another polypeptide chain is there is bond formation, that is your beta pleated sheets. Now this tertiary protein or tertiary structure, this alpha helix can further coil around and the sheeted can further to give you tertiary structure. But in quaternary structures, you have more than one polypeptide chains. Okay, more than one polypeptide chains form a bond to give you a quaternary structure. Okay, so you can see here, some of the proteins are composed of 2 or more polypeptide chains referred to as subunits. Okay, these are your subunits. The spatial arrangement of these subunits with respect to each other is known as your quaternary structures. Okay. Next, explain the classification of vitamins. So we have done with the proteins, now we will go towards vitamins. So classification of vitamins is very simple. You have 2 types, one is fat soluble and another is water soluble. Fat soluble are soluble in fats and oil. They can be stored in the adipose tissue in the liver, okay, and liver. Examples of fat soluble are vitamin A, D, E and K. Now water soluble vitamins are B and C, okay, they are soluble in water, okay, supplied regularly in the diet and cannot be stored in the body. Because they are water soluble, they get excreted during sweating and during urination. So we need to consume vitamin B and C regularly from the diet. Okay, so that is why they cannot be stored in the body. Whereas the fat soluble vitamins like A, D, E and K can be stored in the body. Okay, fine. Next, this is very important, students. So I have not taken any question from here because every year question is asked, but what I have given you is the whole table that you have revised. You need to revise here. Vitamin A, just you need to know the deficiency disease here, okay. It is erophthalmia, which is a hardening of cornea, and you have night blindness, and you should know their chemical name also. Vitamin B chemical names are very, very important. B1 is called as thiamine, okay, and it causes beriberi. B2 is called as riboflavin, and it causes cellulose, okay. Next you have vitamin B6 pyridoxine, it causes convulsions. Vitamin B12 can causes you pernicious anemia. Vitamin C is called as ascorbic acid, can cause you scurvy. Vitamin D can call you rickets, okay. Next you have, vitamin K is also there, that is for clotting of blood. So that table in NCERT book is very, very, very important. Every year 1 marker question. In that 5 marker questions you will have sub parts, right? So from that definitely questions are being asked from vitamins. So please remember the vitamin names, their chemical names and their deficiency diseases. Okay, this is very, very important. Next, coming to nucleic acids, name the type of nucleic acids. You know, RNA and DNA. Name the components of nucleic acid. So I have already discussed nucleic acids. Okay, nucleic acids are the polymers of nucleotides. Are the polymers of nucleotides. Now nucleotides have 3 things. Okay, one is sugar. Okay, one is nitrogenous base, and another is phosphate group. Okay, these are the 3 things present in one nucleotide. So what are the types of nucleic acids? I can write RNA and DNA, right? Name components of nucleic acid. I have sugar. Okay, then I have nitrogenous base. Okay, and then I have PO4 3- or phosphate group. Okay, next, question number 31. Name the type of nitrogenous bases present in nucleic acids. Okay, name the type of nitrogenous bases. Now see, in RNA what are the nitrogenous bases present, and DNA what are the nitrogenous bases present. Okay, so in both of them almost it is same, but there's one difference. So in RNA it is adenine. Okay, same for DNA also, adenine is present. Okay, next we have guanine. Okay, same here also guanine is present. Okay, next we have cytosine. Okay, so here also cytosine is present. Okay, next here uracil is present. Okay, and here it is thymine is present. Okay, so there is one difference in the nitrogenous bases. In RNA uracil is present, whereas in DNA thymine is present. Okay, all other 3 are same. Okay, next, question number 32. What are nucleosides and what are nucleotides? Okay, so sorry, now you can see, first let me answer B, then I'll come to A, do not worry. Nucleotides, okay, as I told you, are the monomers of nucleic acids, okay, which comprises of, or I can write which consist of 3 components, right? What I told you, which consist of a sugar, a nitrogenous base and a phosphate group, right? But what is nucleoside? Now again I'll draw this one. See, nucleic acid, okay, is made up of something called as nucleotides, right? And I told you nucleotides has 3 components, that is sugar, nitrogenous base, okay, and phosphate group. Okay, now the sugar and nitrogenous base alone is called as nucleoside. Okay, so if you add phosphate group to nucleoside, we get nucleotide. Now how is this present? These 3 things, if you have sugar, okay, on the first carbon you have nitrogenous base, we have done an MCQ on this, and on the 5th carbon we have a phosphate group, right? Now what are nucleosides? I can write nucleosides are the combination of sugar and nitrogenous base. Okay, next, question number 33. Give the difference between RNA and DNA. This is also very common and most expected easy question. Okay, so you can just write down any 2 or 3 differences. So RNA and DNA. Okay, so here first of all you can write that RNA is single stranded. Okay, there is only one strand in RNA, whereas DNA is your double stranded. right? Next we can go to their composition. In RNA, the sugar that is present, okay, sugar present is, okay, I have written the difference here, so let me first, okay. Okay, so this is double stranded, this is single stranded. What is the sugar present in DNA? That is deoxyribose sugar. Okay, what is the difference of the sugar present in RNA? It is ribose sugar. Okay, next, you know nitrogenous bases, it is A, T, G and C, adenine, thymine, guanine and cytosine, and here it is A, U, G and C. Okay, adenine, uracil, guanine and cytosine. Okay, these are the differences, 3 points you can write out. Next, question number 34, how many hydrogen bonds are present in adenine and thymine? So here there are AT, right? We have 2 bonds here, and within G and C we have 3 bonds, right? AT like this and GC. Now let us see the next question. Okay, so before going to the next question, the hormones part, okay, we have how many biomolecules? If you see, first is carbohydrates, right? Next we have proteins, okay, then we have your vitamins, then you have nucleic acids, and then we have hormones, right? So we have covered these all things, but hormones I have not taken any question again, but I want to show you this table, you have to remember. So please note down this table, okay. So first of all, how many types, what are hormones? Basic definitions. Hormones are something which is secreted by endocrine glands directly into the bloodstream. Okay, what is their function? They act as intracellular messengers. Okay, this can be asked as a 1 marker question or something. So make note of first these 3 points. Now how many types of hormones are there? Basically there are 3 types. One is polypeptide, amino acid derivatives, and we have steroids. Under polypeptides we have insulin, endomorphins and glucagons. Under amino acid derivatives we have epinephrine, norepinephrine and thyroxine, and under steroids we have estrogens and androgens. Okay, so polypeptide, what we have? Insulin, endomorphin and glucagon. So you can see their functions here. So these are what? These are your polypeptides. Okay, polypeptide hormones category, glucagon and insulin, and their functions are there. Okay, next we have epinephrine and norepinephrine and thyroxine, and these are the types of what? Amino acid derivative. Amino acid derivative hormones. Okay, I am not reading this table in detail, but you know glucosol tries to increase the sugar level, insulin tries to decrease the sugar level, and both of them work together to maintain the sugar level in the blood. Next, if you see epinephrine and norepinephrine, they are responsible for the external stimuli. Thyroxine, very important, questions can be asked from here. So thyroxine is first point, produced by thyroid gland. It is the iodinated derivative of amino acid tyrosine. There is an amino acid tyrosine, and if one of the H is replaced with iodine, we call that as thyroxine. Next, if you have low concentration of thyroxine, that is the situation called as hypothyroidism, and what is the consequences of that? Lethargy and obesity. Okay. Now, if you have higher concentration of thyroxine, it is hyperthyroidism, and the consequence is enlargement of thyroid gland. Okay. Next, we have steroid hormones. Okay. So steroid hormones, again here we have something, it can be produced both by adrenal cortex and gonads. Okay. These are the 2 things which can produce steroid hormones. Now glucocorticoids are released by adrenal cortex. So these 3 things, okay, these 2, one second, yeah, glucocorticoids and mineral corticoids, these are released by your cortex, adrenal cortex. Okay, and gonads, different thing. Okay, so now glucocorticoids, what they do, they are released by adrenal cortex, and they control the carbohydrate metabolism. And what is the role of mineral corticoids? They control the level of excretion of water and salt by the kidney. Okay, now here something you should know, what is addition disease? Addition disease is the malfunctioning of adrenal cortex. Okay, if adrenal cortex is not functioning properly, your glucocorticoids and mineral corticoids will not be synthesized. Okay, in that case, what are the consequences? You can get hypoglycemia, weakness and increased susceptibility to stress. Okay. Next, I told you this steroid hormones can be released by even gonads. So we will see what are the hormones released by gonads. They are your testosterone, estradiol and progesterone. Okay, these are your sex hormones. Okay, which sex hormones means they define the sexuality of a person, whether it is a female personality, male personality, they tell you about the sexuality of the person. So that is why testosterone is released by gonads. Okay, it is a major sex hormone produced in males. Okay, now what is it responsible for the development of secondary male characteristics like deep voice, facial hair, general physical constitution. Now similarly, estradiol is a female sex hormone and which is responsible for the development of secondary female characters and participate in the control of menstrual cycle in women. Okay. And next, progesterone. Progesterone is responsible for preparing the uterus for the implantation of the fertilized egg. Okay. So these are the different hormones, or you can say gonad hormones released by the gonads. Next, we will go to large 5-marker questions. Write the Haworth structure of glucose, sucrose. Okay. So I told you I didn't take you any question from the sucrose because I have taken in this 5-marker question. So Haworth structure is this one. You have alpha D glucose and beta D fructose. Okay, so these two will combine to give you sucrose. Okay. Now let us see the first question here. Now it is 5-marker questions. Explain the Haworth structure of sucrose. So sucrose you can see is made up of alpha D glucose and beta D fructose. Okay, so structure is like this, C1 and C1 linkage is there. Okay, give reason sucrose is a non-reducing sugar. So sucrose is a non-reducing sugar. I told you the trick to find it that you should have a hemiacetal group. Now if you see this is carbon number, in this there is no hemiacetal group, here also no hemiacetal group. So that is why it is an non-reducing sugar. So reducing groups of glucose and fructose are involved in glycosidic bond formation. That is why sucrose is a non-reducing sugar. Next, mention the glycosidic linkage in sucrose or in cane sugar. So C1 of alpha D glucose and C2 of beta D fructose are joined together to give you sucrose. Next, what is invert sugar? Now see, sucrose in general is dextrorotatory, okay, but when you hydrolyze that, it will break to give you glucose and fructose. Dextrorotatory means it rotates the plane polarized light in the clockwise direction. Okay, so now when it undergo hydrolysis, it gives you alpha D glucose and beta D fructose. Now alpha D glucose is dextrorotatory, beta D fructose is levorotatory, right? But what happens here? The levorotatory of the fructose is more than the dextrorotatory of alpha D glucose. So that is why the solution, sucrose was initially dextrorotatory, but when it undergoes hydrolysis, we get glucose and fructose. Levorotatory of fructose is more than glucose, so the solution now becomes levorotatory. Okay, so that is called as invert sugar. Okay. So sucrose is dextrorotatory, but after hydrolysis it gives dextrorotatory glucose and levorotatory fructose. Since the levorotation of fructose is minus 92.4, more than the dextrorotation of glucose. Okay, you can see this is minus 92 and this is plus 52, right? So definitely this one is more. So the mixture is now levorotatory. Okay, so this hydrolysis of sucrose brings about a change in the sign of rotation. Earlier if you take only sucrose, initially it was dextrorotatory, but with the solution, with the hydrolysis, it becomes levorotation. Okay, and the product is named as invert sugar. Clear? Now what is glycosidic bond? The two monosaccharides are joined together by an oxide linkage formed by the loss of water molecule. Such a linkage is called as glycosidic linkage. So you can just write down two monosaccharide units are joined by an oxide linkage by the loss of water, which is called as a glycosidic linkage. Okay. Okay, fine. Next we'll go to the last question. Name the storage polysaccharide present in plants. Okay, we have done a question present in animals, that was glycogen. Storage polysaccharide present in plants is your, present is your starch, right? This is present is starch, storage polysaccharide. Now name the monomer unit present in starch, that is made up of alpha D glucose units. Next, name the components of starch. Starch has two components, that is amylose and amylopectin. Okay, write two differences between amylose and amylopectin. Sorry, we can write two differences here itself. So amylose is what? You can write first of all, it is water soluble. Okay. Amylopectin is water insoluble. Okay. Next, amylose is around 15 to 20%. Okay. And this one is around 80 to 85%. Okay. Here amylose is having C1 C4 linkage, okay, and amylopectin is having C1 C4 and C1 C6 linkage. Amylose is linear, okay, amylopectin is branched. Okay, so these are the differences, you can note down them. Clear? Fine. Now this is, I have made a revision type, okay, where you have just seen all the disaccharides, sucrose, maltose, lactose, their monomer units and the linkage. This is what you should remember. Similarly for polysaccharides, starch, amylose, amylopectin, cellulose and glycogen, what are their monomer units and what is their linkage. So please prepare this table, make this table and learn it again by heart. These questions will be definitely asked from here. Okay. Next you can make this and prepare this. Okay, I have made it for you. Next we have what are the important topics from this chapter. So if you see carbohydrates, glucose, all the 5 reactions, then disaccharides, polysaccharides, that table you should remember. Reducing and non-reducing sugars, their examples and definitions you should know. Next, coming to protein, what is the structure of protein, that is primary, secondary, tertiary, quaternary, their explanations, their examples you should be knowing. Peptide bond formation, Zwitter ion definition and essential and non-essential with example you should remember. Next, coming to vitamins, what are fat soluble, water soluble vitamins, their examples. Deficiency disease is very important. Questions will definitely be asked either in MCQ, this is the favorite topic, so please remember. Next, hormones, you can just prepare about insulin and thyroxine in detail. Okay. Next, if you say nucleic acids, their functions, RNA, DNA difference and the structure should be revised properly. Okay. Next, coming to, one second. Okay, so yes, that's all students in this lecture. So I hope you would find this, and I can say you, if you practice these questions, then definitely this chapter is done for you. So revise it once from our KCET series. You can just revise the concept, then attempt these questions, then you can join our test series which we have launched yesterday. You can join that and attempt questions from these topics. Okay, so this is everything that we are providing you from our channel. If you find these helpful, please do not forget to subscribe the channel, and see you soon in the next episode of Brahmastra series. And yes, I want to tell you the schedule. Tomorrow again it will be physics class, so be prepared. Thank you for watching, my dear students.